Hi students, today we have to discuss about the immunodeficiency diseases, particularly the AIDS. See, the immune system is formed of a number of components, the different types of cells we have seen already, and they normally afford or provide protection to the body against various infections. When the immune system is working properly, there is no infection or anything happens to the body. Sometimes there is a failure in one or more components of the immune system that results in immunodeficiency diseases. We have a number of immunodeficiency diseases because of the defect in the immune system. For example, AIDS, number 2, autoimmune diseases like what we have the multiple sclerosis, myasthenia gravis, Addison's disease, etc. Hypersensitivity, nothing but the allergy, and another one severe combined immunodeficiency disease. Like that, we have a number of immunodeficiency diseases. One such immunodeficiency disease is AIDS. So today we have to give importance to know more about what is called AIDS. So anyway, the immunodeficiency diseases result from a failure in one or more components of the immune system. Now the immunodeficiency may be classified into primary immune deficiency and secondary immune deficiency. What is the reason for the development of primary immune deficiency diseases? So the primary immune deficiency that results due to genital developmental defects. When there is a defect in the what is called the genes or what we call the genome, then it resulted in what is called the primary immune disease. So primary immune deficiency resulted into genetic developmental defects. So when you have primary, ultimately you have the secondary. The secondary immune deficiency is due to a number of causes. It may be because of radiation, exposing the body to the radiations, or use of cytolytic and immunosuppressive drugs frequently. So cytolytic, the one which causes the destruction of the cells. For example, the case of cancer, when you are using cancer treatment, the radiation or any other materials, at times we have developed immunodeficiency. And likewise, use of immunosuppressive drugs. It is being given to the recipient during what is called actually transplantation to prevent graft rejection. For example, one such immunosuppressive drug, cyclosporine. This is immunosuppressive drug. It is often used during what is called transplantation surgery to avoid graft rejection. To the recipient it is to be given. Some steroids also given. When I am using these immunosuppressive drugs frequently, for example the steroids, so why doctors are advising not to take steroid tablets frequently? Because it affects the immune system. So use of cytolytic and immunosuppressive drugs may result in immunodeficiency disease. And also because of infection, sometimes the infection may also cause immunodeficiency. So these are the two types of what is called immunodeficiency diseases, primary and secondary. What are the causes for each one? Normally, AIDS, you know the meaning, acquired immunodeficiency syndrome, acquired immunodeficiency syndrome. So the AIDS acronym can be just elaborated as acquired immunodeficiency syndrome. So it is a disease which is being acquired during the lifetime of an individual, indicating that it is not what we call a congenital disease, not transmitted from. Okay, it is not a congenital disease because it is being acquired. Congenital means from the parents to the young ones, except in the case of pregnant mother. So there is no genetic transmission of AIDS. So because of that one, it is being transmitted or it is acquired only during the lifetime of an individual. Now you know the historical aspect. Two persons, Robert Gallo and Leg Managnia, in National Institute of Health in US, they have identified the HIV virus responsible for the AIDS. This is the thing, you know that one. I don't know, just reveal once again. Now here, we have to study about the virus which causes AIDS. So, the constitutive agent, the structure, the mode of transmission, the pathogenesis, then also the test, then how to prevent it. These are the different categories we have to study under what's called AIDS. Okay, so the causative organism for AIDS is HIV virus. HIV, that is what we call human immunodeficiency virus. Formerly it had different names. Now finally we have the name static now used frequently, human immunodeficiency virus, because it causes immunodeficiency. Now this virus selectively attacks one specific type of cells, what is called the T cells. So the main cell which is being affected by this virus is T cells, particularly T lymphocytes, otherwise called one type T helper cells. T helper cells. So it is one type of what is called T lymphocytes. We have many types of lymphocytes. We have seen already B lymphocytes, etc. But this virus selectively attacks the helper T cells. And once they are being attacked, the T helper cells are responsible for stimulating the B lymphocytes to produce antibodies. So once these cells are attacked, the T lymphocytes fail to stimulate the B lymphocytes to produce antibodies. So that results in decreased number of T lymphocytes because the T lymphocytes are affected. One such type of T lymphocytes affected by the HIV virus is CD4. This is one type of what is called T helper cells, CD4. This is a cell normally affected by what we call the virus. So it selectively destroys the T helper cells. Because T helper cells are responsible for the stimulation of the B lymphocytes to produce antibodies. Now what is happening? There is a loss of what is called natural defense against the viral infection. Loss of natural defense against the natural viral infection. Loss of natural defense we can see. Loss of natural defense against the viral infection. Now we come based on the genetic characteristics and also based on the differences between the antigens. There are two different types of HIV. HIV-1, this is one type. HIV-2, this is another type. This is a classification based on the characteristics, genetic characteristics, and also the difference between the antigens possessed by the virus. That is about the types. So this is what we call the introduction part. We have a number of what is called immunodeficiency diseases because of the failure of the immune system. Now we are taking first one, the AIDS, and what is the constitutive agent, what is the structure of that agent, how is it transmitted, how does pathogenesis occur, what are the tests used, and what are the preventive measures. These are all things we have to go through now. Now let's see the structure of HIV. Now this virus is an example for enveloped virus, just like hepatitis B virus. Other viruses are without envelope. Because of the presence of envelope, the envelope is a lipoprotein structure. So we have a lipid bilayer present outside. Now so that the virus is sensitive to lipid solvents, like for example benzene, chloroform, ether, etc. So what is the purpose of using sanitizer nowadays? To just actually to prevent the infection from coronavirus, because the envelope actually, so the organic solvents can dissolve the virus, like benzene, chloroform, ether. These are all the components present in the sanitizer. That's why we are using what is called actually sanitizer, just for cleaning the hands to remove the virus. So enveloped virus sensitive to lipid solvents like benzene, chloroform, ether, etc. Now it belongs to the genus lentivirus, an example of retrovirus. The meaning for retrovirus, the virus has an enzyme reverse transcriptase. So having the ability of undergoing reverse transcription, hence called retrovirus. Now the virus is normally spherical in shape. Then the size is 100 to 120 nanometer. Now if you are taking the components, it is having dense inner core. This is the dense inner core. And the outer lipoprotein envelope. So this is the envelope, there is a red one and the blue one, both together called as lipoprotein envelope, form of lipid bilayer. Now this is the lipid bilayer, and also we have this is the protein. The protein. That's why it's called lipoprotein envelope. Lipoprotein envelope, formed of both proteins and lipids. Now attached to what is called actually the lipoprotein envelope, we have spikes of glycoprotein. Now this is what we have, the spikes of glycoprotein. Just lead-like structures, just like the what is called the component of the pin, having a head-like structure, what is called the knob, and then a needle-like structure. Now we have GP sticks out inside and outside position of envelope. You see that one, if you are taking each GP, it sticks out inside and outside of what is called the lipoprotein envelope. Now the outside position of what is called the spicule, the spikes, is made up of what is called glycoprotein GP120, somewhat smaller molecule. And the inner position is called GP41. So this is a surface one present on the surface, having a knob-like structure. That's why it's called the surface proteins. And the one which is now passing through the lipoprotein envelope, hence called transmembrane protein. So it is of somewhat larger when compared to GP120. So it is GP41. That refers to what is called the size of the particles which get sedimented. So GP120, GP41 are the two components of what we have the spikes. Now the GP120 outer surface protein, GP41 the transmembrane protein passing through the envelope. Now it is a GP120 which is being attached to the surface receptor of what is called the host cells. The contact between what we have the host cells and the virus occurs mainly through what is called GP120. The GP120 of the virus normally attaches the virus to the surface receptor of what is called the host cells. Particularly we can say the T lymphocytes. Okay. Now what are the components present in the inner, what is called the dense core? The inner dense core has a capsid protein. Capsid. That is what is called the outer covering. It is normally icosahedral in shape. The capsid, inner capsid. That is, it is normally icosahedral in shape. Icosahedral. Icosahedral in shape. That is about the inner isohedral. Inner, what is called the capsid. That means it is having 20 faces. 20 faces, just like the diamonds, we have the capsid, the outer, that is the layer of the inner core, the dense core. We have a capsid core made up of protein. Then it is having 20 faces. If we make the difference between the capsid protein and what we have the matrix protein, this is what is called P17 and this is what is called P24. P refers to the protein. The just a symbol is for what we have the different types of proteins according to the density, etc. So capsid protein is P24, the matrix protein, the outer one is P17. Now this capsid, this is now called as nucleocapsid. Nucleocapsid. Entirely called what we have nucleocapsid. Because it is having outer capsid layer and also inner nucleic acids. Now it is an example for diploid virus. It's an example for diploid virus because we have two different or two strands or two separate strands of RNA. That means two single stranded RNA. It's not what we have that is double helix. They are present separately. Hence they are called single stranded RNA. So we have two molecules of single stranded RNA, hence the name diploid viruses. Now attached to this one, we have what is called actually enzyme. Enzyme are present attached to that is what is called this one, and this is called the reverse transcriptase. So reverse transcriptase enzyme is attached to what is called this single stranded RNA, and both have the attachment of reverse transcriptase enzyme. This enzyme is normally responsible for the reverse transcription process, hence the virus is belonging to retrovirus group. So two separate single stranded RNA with what we have the molecules of reverse transcriptase enzyme attached to them are present. In addition to that one, we also have different enzymes present in the inner core. Say an example of protease. One enzyme we have protease. So it is normally P10, you can say protease. So this one, what we get the reverse transcriptase is also what is called enzyme protein. This is what we have P64. The reverse transcriptase enzyme is P64. We also have some more enzymes what is called integrase. Integrase also present in the inner core. So it is of P32. Like that we have different proteins. Also in the inner core, along with what we have, that is enzymes, proteases, reverse transcriptases, integrases, we also have the enzyme ribonuclease. That enzyme is also present inside. So out of this cancer only we have what's called the protein matrix. This is called the protein matrix, the blue one, and it combines with the lipid layer, the bilayer, what is called the lipid of outer, to form lipoprotein envelope. Okay, that's about the structural aspects of actually lentivirus, what we call HIV. Right? Now the attachment with what we have the receptor of the host cell is by GP120. Now normally the virus is looking like a soccer ball. The surface protein is formed up, so it is looking like a soccer ball. So it is formed of 12 just what is called pentagens and 20 hexagons. So pentagens, you know that one having 5 6, just look at the surface of the football, we have just visit that in black and white. It's not a single leather, it's being formed of many pieces of leather. So just like that one is being formed of 12 pentagens, 5-sided, and 20 hexagons, 6-sided pieces of proteins combined together to form what is called the virus. How far these viruses transmitted from one individual to another? That is, what is the mode of transmission? So the main mode of transmission is unsafe sexual contact. Unsafe sexual contact. Normally the virus is formed in the living cells, particularly the human body, the macrophages. It is multiplying more and more in the macrophages only. Hence the macrophages together called HIV factory. So the HIV factory is nothing but the macrophage. Now what are the routes of transmission? I mentioned about the first one, unsafe sexual contact. Now the second one, blood contaminated needles, as in the case of persons with the drug abuse. The same needle has been used by different persons in the case of drug abuse. So blood contaminated needles. Then blood transfusion from one person to another person without screening the blood, while transferring the blood from one person to another, that is from the infected person to another without screening. That is also one of the methods. Then also during what is called organ transplantation, or what we call the graft. The grafting between one to another. Suppose a person is infected. If his organ is transplanted to another normal individual, he will get HIV, that is in his body. Then vertical transmission from infected mother to child. I already mentioned horizontal gene transfer. Vertical transmission means from one generation to another generation of the same species. Horizontal transmission means from one species to another species. That's called horizontal transmission. Here the vertical transmission occurs from the infected mother to child through placenta during pregnancy. So vertical transmission from infected mother to child during pregnancy through placenta is also one of the methods. It's a rare method. Okay, that is from infected mother too. That's called vertical transmission. Horizontal transmission from one species to another species. Vertical transmission from one individual to another individual of the same species, from one generation to another generation. Now, what are actually the factors which normally do not transmit the virus, generally speaking? Insects are not transmitting the virus. Even the casual contact with the infected person will not transmit the virus. Casual contact, not actually intimate contact. Casual contact also doesn't transmit the virus. So that is about the mode of transmission. We have to go for other things, the pathogenesis, etc. Now the next aspects of disease, that is pathogenesis. It refers to the development of the disease. What are the different stages that occur during the development of the disease? So first the viruses get into the cells, particularly enter the macrophages. Inside the macrophages or in the macrophages, the viral RNA genome replicates. That is to form viral DNA. The genome, that is normally RNA, it gets converted to form what's called viral DNA. This is by a process what's called reverse transcription. Normal transcription, DNA directs the synthesis of RNA, but here the RNA directs the synthesis of DNA, hence called reverse transcription process. It is brought about by the enzyme reverse transcriptase. That is nothing but actually, you see that one in this process, RNA dependent DNA polymerase. It is RNA dependent DNA polymerase because it synthesizes viral DNA. Now, what is happening? In the macrophages, the virus particles continue to produce more and more virus particles. So in this way, the macrophages produce large number of virus particles. Hence, it is aptly described as HIV factory. When it is going on inside the macrophage, where we have the production of varying number of virus particles, at the same time, the virus particles also enter into the T lymphocytes, particularly the T helper cells. It is a switch on for the B lymphocytes to produce antibodies. Now, the virus particles not only enter into the macrophages but also enter into the T lymphocytes, where replicates and produce many virus progeny particles or many progeny viruses. Now by the outburst of the T lymphocytes, the virus particles are released into the blood. Now they are attacking their fresh T lymphocytes. As a result, what is happening? Decrease in number of lymphocytes occur. The decrease in number of lymphocytes make the individual, make the infected person, that is, starts suffering from infection, and also the person becomes immunodeficient and unable to protect himself from infection. So these are all the different changes leading to what we have, the AIDS disease. So we have what is called normally to begin with various symptoms. And finally, we have AIDS-related complex develops, ARC, AIDS-related complex. That's not necessary. So these are the various stages that lead to the development of the disease, and that process is called pathogenesis, the development of the disease. Now to see what are the symptoms that develop during these periods when we have the burst of the T lymphocytes, decrease in number of T lymphocytes, making the persons unable to protect himself from the infection, suffering from, that is what we call infection, etc. Now see the symptoms related to the AIDS. What are the symptoms of AIDS? 1. Bouts of fever. that is a recurrence of fever, that is persistent fever, chronic fever. Then weight loss, at least 10% of the body weight is lost. Then diarrhea, chronic diarrhea, just occurring continuously. Then persistent cough. Persistent cough also just a dry cough without cold, it's also persistent continuously. Then oropharyngeal candidiasis, a kind of fungal infection in the mouth. We have seen already fungal infection candidiasis, that is fungal infection in the mouth. And oropharyngeal candidiasis means fungal infection in the mouth, normally called thrush. Normally called thrush. So this is fungal infection, common name thrush, candidiasis in the buccal cavity. A peculiar type of what is called sarcoma in the connective tissues, what is called Kaposi's sarcoma. This is normally having symptoms, the sarcoma. But in the case of AIDS patient, it has different symptoms. It causes what's called complex patches on the chest region and also the neck region and also in and around the mouth. That is Kaposi's sarcoma, a kind of cancer in the connective tissues. Loss of memory and intelligence. As the brain cells are affected, loss of memory and intelligence to occur. Then opportunistic infection. So normally the tuberculosis is waiting for a chance to enter the body when there is a defect in the immune system. As it is an immune deficiency disease, allowing more and more what is called the bacteria, the one which cause, that is tuberculosis. As tuberculosis is waiting for a chance to enter the body when there is a loss of what is called the immune system, when there is a deficiency of immune system. Hence it's called opportunistic infection, waiting for the chance to enter. So that is what we have the tuberculosis. So these are all the different symptoms we have. We have more symptoms also. But I have to restrict only a few symptoms as per the book. Even in the book only just 3 symptoms are given. That is bouts of fever, weight loss and diarrhea. All the symptoms also added just for your reference because some questions related to Kaposi's sarcoma, that is in NEET question paper. A type of cancer related to AIDS is called Kaposi's sarcoma. Now, diagnosis of AIDS. A simple blood test is available to just actually find out whether the person has been infected with HIV or not. Now, the simple blood test is available, I mentioned. What for? This is a biochemical test. It's not being used to find out the presence of HIV virus. The test is used to find out the presence of antibodies against the proteins of the HIV. So it's a biochemical analysis, even also called the color test. Now simple blood test is available to detect the presence of antibodies, not the antigen, to detect the presence of antibodies against P17 protein of matrix. There is one protein what's called P17 present in the outer layer, just present nearby the bilayer of lipid matrix, and P24 proteins which make up the capsid. The inner nucleic capsid, the capsid is formed of one type of protein what's called P24. The outer just envelope, below the lipid bilayer we have the matrix protein that is formed of P17. So these two, when virus is present, now these two proteins are acting as antigens. So they stimulate the production of antibodies. They stimulate the production of antibodies. By determining the presence of antibodies, one can able to find out whether the HIV virus is present or not. Now this P17 protein is present in all the retroviruses, but the P24 is present only in HIV. So P17 is present in all the retroviruses, enveloped viruses. But the P24 protein, the capsid protein, is found only in HIV, not any other. So if you are detecting the antibodies to this protein, that is a confirmatory test. Okay. So two tests are available. One, ELISA test. enzyme-linked immunosorbent assay test. I will explain the mechanism of ELISA test under biotechnology, applications of biotechnology in the next chapter. What is the method adapted in ELISA? Now it is a preliminary test because I mentioned already P70 is present in all the retroviruses. So it is used to detect HIV antibodies to proteins, that is P70 in the outer core. When this protein is present, it stimulates the body to produce antibodies against this protein. So we can find out the presence of antibodies. Now another one, the Western blot test. Southern blot test is used in DNA fingerprinting. Here the Western blot test. Just the blotting mechanism is adapted as in the case of DNA fingerprinting. It is called Western blot test. It is a reliable and confirmatory test because I mentioned this P24 protein is present only in HIV, not in any other viruses. So we are detecting the antibodies to this protein. When this protein is present, it stimulates the production of antibodies against it in the blood. So by means of biochemical analysis, one can find out the presence of antibodies against this protein. Okay, so based on detecting antibodies to viral capsid protein, this is outer envelope protein and this is what is called viral capsid protein, otherwise called nucleocapsid. Okay, so this is what is called the confirmatory test, reliable test based on detecting antibodies to viral core proteins. If both the tests detect the presence of antibodies in a suspected person, then the person is said to be positive to HIV or positive to AIDS. So if both the tests detect the presence of antibodies in the suspected person, he is normally considered as HIV positive person or AIDS positive person. That's about the diagnosis of AIDS. Now prevention and control of AIDS. There is no cure for AIDS, but the prevention is a better option. What are the preventive measures? So prevention is always better than that one, that is the disease. So advocating safe sex contact and promoting regular checkup whether they have AIDS or not. So safe sex contact, that is the main one. This is a primary, that is what is called prevention. The use of condoms during the sexual intercourse or during the sexual contact. Then another primary, just actually prevention is safe blood for transfusion, screening the blood before the blood is to be transfused. Screening means whether HIV is present or not. The use of disposable syringes. Normally you know that one. The disposable, if you are using the syringes already used, it is very common in the case of drug abuses. They only use the same syringe for multiple persons. That will transmit the disease through contaminated blood needles. So use of disposable syringes, one of the methods normally. Actually, because of syringes, what is called the needles, blood contaminated needles. So you have to use only one time the syringes for the people during injection. So use of disposable syringes, because syringes are transmitting the disease. If you are using the same syringe for different persons, then prevention of drug abuse. Because drug abuse is nothing but taking what is called the abnormal drugs. For example, just we have even, I will also consider as a drug, morphine. We are going to study the next one about the different types of what is called ganja, abin, etc. So prevention of drug abuse is one of the methods adapted to prevent what is called the AIDS, particularly in the case of young generation people. Then awareness by different organizations. Number one, NACO. So National AIDS Control Organization, even run by the government. NGO, other non-government voluntary organizations. And World Health Organization also frequently they are delivering just the methods how to prevent the disease, just like you see that one COVID-19. So World Health Organization, so they are creating in the mind of the people about the importance of what is called just preventing the disease, the different methods. That's about the awareness. Now every year December 1st is actually considered as a World AIDS Day. We cannot say celebrated, but it's not a celebration, just to remember. and just make awareness in the mind of the people. So December 1st every year is considered as a World AIDS Day. So that's about what I concluded about the various steps of what is called this immunodeficiency disease. The structure of HIV, the mode of transmission, the development of the disease, the test, and also finally how to protect the body from AIDS, the preventive measures. That's all about AIDS.